Another A-level biology video, and this is Mr Phillips, and we're going over DNA, chromosomes, genes, and all sorts today like that. So within this lesson, hopefully you'll be able to define the term gene even, and explain how DNA is stored in different cell types and organelles, explain how the DNA codes for amino acids in protein synthesis, explain the difference between introns and exons. So let's have a look at this then. So first of all, I want you to have a go at this task if you haven't done it. So just pause the video and then we will go through it together in a few moments. Okay, so let's actually have a look at this, the answer to these questions to make sure we're happy, because until you can do these, you can't really move onto the other sections of this module. So first of all, what three places is DNA found in eukaryotic cells? So most students will know that they're found in the nucleus, because obviously that's where your chromosomes mainly are of each cell, but it's also found in the mitochondria. Okay, so there's DNA in your mitochondria, and there is DNA in the chloroplast, which we're talking about in a few minutes. So that is the other two places it can be found in eukaryotic cells. That's question one done. What four bases make up the DNA structure? Well, hopefully you know that the base is part of the nucleotide, which comes up in a later question. But effectively, you've got your four bases over here in this picture. So we've got A, T, G, and C. So that's adenine, thymine, guanine, cytosine. And for A level, you need to know how to spell those words correctly. What does the complete base pairing mean? Well, again, that picture again shows you that it's always the same base pairs. So A to C, G to C. Question 4, what links these base pairs in their complementary base pairs? So they're hydrogen bonds, but you need to know that for A to C, it's always A to T, so it's always 2, G to C, it's always 3. So A to T, 2, G to C, 3. Describe a DNA nucleotide. So basically, you need to know it to this sort of level only up here. Okay, so you'd be expected to say a DNA nucleotide is made up of a phosphate group that is connected to a pentose sugar, and if it's DNA, it's deoxyribose. If it's RNA, it's ribose, and that is attached to a nitrogen-containing organic base. Next one, describe, so we've done that one, sorry. The next one, number 6, what is an antiparallel mean? Well, if you look at the diagram, it shows you what antiparallel means. Antiparallel means that the direction the sugar molecule is facing. So one sugar molecule is facing upwards or downwards, depending on which way you're looking at it. And then number 3, how would you know that the images are from the two pictures below, which are a chloroplast and mitochondria? How do you know they've been taken using a transmission electron microscope? Well, you would know because they are 2D images. Okay, so what we're going to look at now is how DNA actually makes up the chromosomes. But if you don't know the structure of DNA, it'd be well worth following the video on here, which will be on the link available on the actual PowerPoint, which obviously I'll have sent you. That would be really, really useful for you. If not, however, just move on to the next. What we're going to be looking at today is how DNA is stored, where it's stored, how it's what structure actually is, because it isn't just found in our cells as just DNA. So you need to know that the structure of DNA, the antiparallel double helix, is the same in every single organism. However, it is stored differently. So if we have a look, you can see in eukaryotic cells, which obviously animal, plant cells, fungal cells, okay, the DNA molecule is a linear structure. So what that means is it's a straight structure, and they're formed obviously in chromosomes. The DNA molecule is too long, so it wouldn't actually be able to fit in your nucleus of your cell unless it was wound really tightly. And the chromosome is a strand of DNA that is wrapped around a histone protein. So if you imagine you've got a histone protein, which would be like that, that would be a histone protein, and then your DNA is sort of wrapped around it as tightly as it possibly can. Okay, and then if we could store, if that was a Y, you'd be able to store lots of wire wrapped around something. and that means it can be fitted in there. The chromosomes are very compact, and the DNA is extremely tightly coiled around the histone protein. However, there is also DNA in your chloroplast. Here's an actual map of some chloroplast DNA, which is easy to follow and actually to map out the histone of it. So DNA is found in the chloroplast of mitochondria, but it's actually similar to prokaryotic DNA. It is naked DNA, so it's not associated to histone protein, and it's much smaller lengths of circular DNA. So very similar to sort of plasmids and chromosomal DNA. Prokaryotic DNA. So if we do look at prokaryotes, so we still need to understand that prokaryotes still have chromosomes technically. It's naked DNA, so the DNA is shorter and circular and is not wound to a histone protein. However, it does supercoil to fit into the cell, and that's why we still call it a chromosome as opposed to just saying it is just DNA. So you can see here, you've got the circular bit of DNA, and then that supercoils up to make it take up less room. And you can see that's actually happening during this this part here that's called on the electron microscope. So you see it's supercoiling there. So for this module, what we're going to be looking at and particularly covering, how does DNA code for proteins, and what does the DNA do in this job? Well, the first thing you need to know is what the term of a gene is, and this is really, really important that you learn this. So I've sort of given you this several times before, but this is the exact definition that you need to actually know and take forward to your A-level exams. So a gene is a sequence of DNA bases that code for either a polypeptide or functional RNA. And we'll worry about what functional RNA is in the next few minutes. But for the first few minutes, we're going to look at how does it code for a polypeptide. So you should understand what polypeptide is by now. You should understand that polypeptide is a strand of amino acids that makes up your protein, further folded into proteins. And in order to make that strand of amino acids, which makes the primary structure of the protein, we need DNA. The first thing is, if you look at this picture, it shows you quite nicely that there are things called codons of DNA, okay, which may also be called triplets. So codons and triplets mean the same thing, and all it means is every 3 bases of DNA, okay. So if we have here, for example, the codon GAG, so guanine, adenine, and guanine again, okay, GAG will be copied, and this length of DNA, this gene, and when we do protein synthesis, only one gene is copied, not the whole DNA. This gene is copied into messenger RNA in a process called transcription, which we'll look at next lesson. What that does is it makes a length of RNA, or messenger RNA as we want to call it, okay, which is going to be the length of one gene. And as you can see here, it's got lots of sets of codons. So you've got CAG, CCC, UCA, AUC, etc., etc., etc. Now, those 3 bases, those codons, every single triplet, every codon codes for 1 amino acid, and they'll always be the same. So, for example, GAG will always code for glutamine. Okay, so that therefore doesn't change, it's specific. So summarizing, every 3 bases in DNA is a codon. 1 gene of bases in DNA is copied into 1 gene of messenger RNA in transcription. Every 3 bases codes for 1 amino acid, and therefore the order of codons codes for the order of amino acids. And how do we make it? Well, there's an example which we'll look at again, of no doubt at some point, but making proteins from DNA triplets, you can see here, or from codons, you can see here the code. We know what different codons make different amino acids. So for example, if I had the code U, let's do it randomly, G and A. What that would code for, if I look on here, so go to the middle section, which is where I start. If I can find my mouse, sorry. I go, right, U. There we go. Oh, I've chosen a bad one. G. And then A there. And that actually stands for something called a stop codon. So it's a pretty bad example. So if I did another one, let's say UGC, it would code for cysteine here. Okay. We'll talk about what stop codon is later on. So that's how we code for proteins. But if you look at this, the definition of a gene, it is a sequence of DNA bases that code for either a polypeptide or functional RNA. So we need to know what functional RNA is. We don't need to know how it's coded for, particularly how it works, this one. We just need to understand what it is. So functional RNA, now looking at the blue box, is not messenger RNA. So it's a different type of RNA other than messenger RNA. So that's quite important to know that that's what functional RNA is. So it's not messenger RNA. Okay. It's involved in performing special tasks during protein synthesis, and examples of it include transfer RNA or tRNA and ribosomal RNA or rRNA. And we'll talk about those a bit more in the future. But effectively, all you need to know is that a gene code is a sequence of DNA bases that codes for either a polypeptide, which will make a protein, or functional RNA, which is RNA that is involved in the production of proteins in protein synthesis. And two terms that you also should hopefully be familiar with. The first one, a genome is a complete set of genes in a cell, but a proteome is the complete set of proteins that a cell can produce. Because obviously a cell might have a full set of DNA, but a lot of the genes are turned on and some of the genes are turned off. So proteome is the complete set of proteins a cell can produce. So that seems all straightforward, but actually there are areas of DNA that are non-coding DNA called introns, and they don't code for proteins or polypeptides, and they don't code for functional RNA. We don't actually know the purpose of introns because they don't exist in prokaryotes. So most things that are important started in prokaryotes and continued into eukaryotes. These have appeared in eukaryotes. So what possible use could they have? Well, we don't really know, but we do know what we do to them to remove them when we're doing amino acid, making our proteins in amino acids in translation. So what happens is this. So if you took a length of a gene, and the gene would have bits of exon, okay, which are the coding regions, and then bits of intron in them. So we want to get rid of these red intron bits, the non-coding regions, because they're going to disrupt our sequence of amino acids if we've got these big blocks that don't actually code for anything in the DNA. So the DNA, nothing happens to, but don't forget the DNA in the process called transcription, which we're going to talk about next lesson, goes into makes something called messenger RNA. And first of all, though, we have something called primary RNA. Now, primary RNA contains both the introns and extrons. It contains the introns, the non-coding bits, and the exons, the coding bits. And a process happens called splicing, which we don't need to know the details of, but it'll involve enzymes, something called endonuclease enzymes. And what they will do is they will effectively cut out the introns, the non-coding bits, so you're left with just blue exons here. And then your amino acids can be coded for, as there's no gaps in the code effectively in the messenger RNA. Also, if between genes there are lots of what we call repeating units of DNA, so where the bases might keep repeating. So for example here, and the picture is really irrelevant, it's just to give you an example, we've got a repeating code of AGAT that occurs here 7 times and here 6 times, and that is called a non-coding multiple repeats. So outside the gene there might be big gaps of sequences where you have lots of non-coding regions of DNA, and that really much might be to split the gene up, but also again we're not always 100% certain what the role of that is. So here we have something new to start to look at then. So we've got alleles. So you need to know what an allele is, and it's a different form of a gene. You might have said at GCSE it's a version of a gene, but basically the base order is ever so slightly different, and that is caused because of a mutation. Okay, and that is caused due to have a different version of the same polypeptide. So it still makes the same protein, but it might have a very, very slight difference to it. And we see these alleles occurring at somewhere called a locus, which is the position on the chromosome. So if you have a look here, this is a simplified version, but you've got eye colour here, and you've got two slightly different versions of green eye colour, but they're found in the same place on the chromosome there. You've got blood type, which could be different types of blood types and different alleles of that, but again found in the same place. Hair colour, same thing, and growth links out away. Okay, and basically these are found on the chromosomes, and these different alleles lead to the genetic crosses, which you'll do again in year 13, which you'll have done in GCSE. So you have different versions of the same gene, and therefore you might have inherited these are what we might call homologous chromosomes. So this one might be, from your, I'll just do an M, from your mom. This one might be from your father, and therefore you have different versions of the same gene inside your cells. Now, this could lead to a question, okay, which we're not going to look at massively yet. I'm going to keep reintroducing this question to you to help you over the next few weeks or lessons. But it says the yellow colour in peas is the result of the enzyme that breaks down chlorophyll, which is green. The Y allele codes for the production of the enzyme that breaks down chlorophyll. The Y allele is a result of mutation of the big Y allele, so you've got a dominant recessive one. The recessive Y allele codes for the inactive form of this enzyme. Outline how the Y allele codes for the production of this enzyme and explain why the recessive Y allele codes for an enzyme with a different primary structure. So you need to just be aware, and we'll only just really briefly mention this, that an allele is caused by a mutation. A mutation is a change in the gene sequence, a change in the DNA basis. So this one's got GAG, and it codes for the normal phenotype, which is represented as yellow here. This one has CTG, so that A organic base in the middle has been substituted for T, and that causes a change in colour of the person there. And that's all an allele is. It's mutation that leads to a change in the what we call the phenotype, the characteristics you show. I don't want to look at this massively for the minute, just this top section for me, because it's the only bit we've just done. So you've got the DNA sequence codes for the amino acid sequence, and also obviously the messenger RNA sequence in the middle. Every 3 bases or triplet codes for one amino acid, and we'll talk about what degenerate code is in a moment. A mutation has happened which changes the DNA, and therefore it could change amino acid, but it might not, as we'll talk about at some point in the future. Just to introduce that question, we'll look at that a bit more in much more detail in a couple of lessons time once we've covered all this content. So then we've got this question here, so this picture here. So just to introduce then, so we've talked about alleles. We just want to introduce the term homologous, okay? So this is the idea that you have 46 pairs of chromosomes, 23 pairs of chromosomes in your body, in every cell in your body. You have one of those pairs of chromosomes, one of those homologous chromosomes has come from your dad, and the other one has come from your mother. And therefore, you have 50% of each chromosome inside your body. That's called a homologous chromosome. So one from your mom, one from your dad, which have different versions of the same gene, different alleles of the same gene. And we've talked about the term diploid and haploid. When you've got 46 chromosomes or 23 pairs, we call it diploid. When we make egg or sperm cells, your gametes, it becomes haploid because you only have 23 chromosomes rather than 23 pairs. Now, the last bit is just to introduce this term of DNA bases and DNA sequencing. So just so you're aware really with this is that the DNA code is known as a common degenerative non-overlapping code. So what does that mean? So you need to know what each of those terms means. So you should know now, because I've said it loads of times, that a triplet or a codon is 3 bases that code for one specific amino acid. Some triplets code for stop codons. So when I went through that example earlier, I chose a bad example in some respects to try and teach it you, but I ended up doing, I think it was UGA, which came to this red stop. And a stop codon is basically when you get to the end of a protein, you need something, or the end of a gene, you need to tell the RNA polymerase or the DNA polymerase to stop doing transcription at that point, and that's what stop codon does. It's like a full stop to the protein, effectively. Basically, these are the same codes in every single living organism. So the code, let's have a look here. The code UUA will always code for leucine. It doesn't matter what organism it is. So it's common across all living organisms that we are aware of. Next term is degenerative. So alanine is coded for, so this one circled in red there, is coded for using the following different triplets. So it could be coded for anything being with a G, anything then goes with a C, but it could be GCU, it could be GCC, GCA, or GCG. Okay, and those 4 different base codons all code for alanine. So it doesn't matter if we get any of those, it will always code for the same one. And this term is called degenerative. In fact, it has more than one messenger RNA codon that codes for the amino acid. Okay, and next one is there are non-overlapping. So what that means is each amino acid is coded for by 3 bases. Ribosomes translate a whole codon at a time, so they never overlap. They look at each 3, then the next 3, then the next 3, then the next 3, and that is a non-overlapping code. And it's worth pointing out that this here is the codons on the messenger RNA, and that's why we have U here, not T, because this is the copied codon, the copied messenger RNA code, which has come from the DNA. Okay, so just sort of finishing off then, we've said that we haven't said this yet, but there are start codons, okay, and they are always involve AUG. So if you look, really, there's only one green start codon here, and this starts all amino acids. So A, then you've got a U, then you've got a G, which gives you your start codon, okay, which codes so-called thymine, which starts all amino acid or all protein sequences. And it will always have a letter A, 3 bases in front, so the X could be anything, but we've got a letter A, and it always is followed straight away by a G, okay, and that would be called what we call a start codon, so it starts all amino acid sequences, or all genes, I suppose, as a result. Okay, and then just to finish off, hopefully, but yeah, just to finish off, here's an exam question. Now, obviously, you would have the something to help you here. You're not expected to remember the different bases. So you'd have something like a wheel, like I just showed you, to produce this. But effectively, you could get a question here where you're asked to look up what the AUG is, what AGG is, and just use information given to tell you what amino acids have been coded for. Okay, guys, so that's an introduction to DNA structure, chromosomes, introns and exons, alleles, and then the DNA code, the genome. So I hope it's been useful for you. Please go back, please use it. You really need to make sure you understand everything that's being covered in this video. Thank you. See you again soon.